Good morning, professors. I am Dr.G. VARATHARAJU, Assistant Professor, Department of Botany, Sri Kaliswari College, Sivakasi. So once again, I feel happy to express my thanks to the organizing committee. In this lecture, we'll see about the sequence analysis. What are the tools available for sequence analysis and the application of sequence analysis under the heading Bioinformatics tools and applications. So this slide shows the significance information related to the genomics and the proteomics. The genomic research starts from the analysis of the genome data. So if we have the genome data again converted into amino acid data. So the amino acid data, from the amino acid data, it can be processed for different purposes. For example, from the amino acid sequence, we can construct 3D structure of the protein. Or if we want to know the motif of the protein, it is possible with the help of sequence analysis. And if we have amino acid sequence, from this sequence, we can analyze the relationship among the organism, how or protein level or organism level, how far it is closely related or how far it is distantly related. And in pharmacology, we can analyze the protein, whether the protein can be binds with or interact with a particular drug or other the proteins or hormones. So it is very useful for the drug development field. So the discovery or the isolation of or retrieval of gene or genome sequence is very important. So with this slide, I will continue the importance and the tools available for sequence analysis. For each and every work we do in bioinformatics, the work starts from sequence. Without sequence, it is very difficult to carry out any kind of bioinformatics research. So that is why I have framed this slide to show how the sequential data were evolved in different period of time. So in the year of before 1955, we had or we know the structural information about the DNA. So the great scientists Watson and Crick explained about the double helical nature of the DNA structure. So after the Watson-Crick discovery, so we know that the genetic material has a double-stranded in nature. And after 1955, sequence technology were developed, Sanger sequencing. It was used to sequence the insulin protein. So by this time, we know how the molecules are sequenced. So from this research, one by one, the technology were developed. So after getting the sequence, we have developed tools for aligning the sequence. And finally, we developed databases. So already previous class we discussed about the biological databases, the significance of biological database, types of biological database, and the applications of biological database. So the biological databases are developed after 1970 or in the beginning of 1975. And finally, there are a huge number of databases are available. The purpose of developing the database is the scientist to know the storage of sequential information is very important. So since the scientist aware of the storage of sequence data, the scientist developed databases. There are two different databases, the nucleotide database and the protein database. So in order to avoid the unwanted confusion, they have developed separate databases. And also the nucleic acid, the physical chemical property of the nucleic acid is entirely different from the physical chemical property of the protein. So that is why the databases are separately developed, and it is stored based on the biological nature of the sequence, I mean nucleotide sequence and the protein sequence in separately. And for DNA sequencing, Sanger DNA sequencing method is used. So it is developed in the beginning of 1980, and in the year during 1985, we have developed the PCR PCR technique. So the PCR technique was very useful to amplify the gene. So amplification is very very important, because if you have the sufficient amount of a gene product, then only sequencing can be possible. So otherwise we'll get the poor result from the sequence. So that is why here the PCR polymerase chain reaction is considered as most important instrument for sequencing the DNA. Okay, so from this knowledge, we will sequential sequence analysis. The sequence analysis is very very important for the genomic research and proteomics research. There are 5 different methods is regularly used for sequence analysis. They are knowledge-based single sequence analysis, pairwise sequence comparison, multiple sequence alignment, sequence motif discovery in multiple alignment, phylogenetic tree. These 5 methods are used for different purpose. The comparison method or alignment method also different. What kind of method will be useful? That is different scenario. The method is used based on the purpose we are taking care. What purpose, what we need, based on that we can use any one of the sequential analysis method. For example, pairwise sequence alignment or comparison. So if you have 2 sequence with you and you don't know anything about one sequence, but you have complete information about one sequence, the second sequence you don't have anything about the second sequence. So in this case you can do comparison between the 2 sequence, whether the each sequence are closely related or distantly related. So whatever the information present in the first sequence, if the same thing is present in the second sequence also, we can conclude that both are closely related. Otherwise we can conclude that both are distantly related. So it is since the alignment process is carried out between 2 sequence, so that is why the method is called as pairwise sequence alignment or pairwise sequence comparison. The pairwise sequence comparison can be performed either with the help of a sequence, or we can perform this method to structural comparison. Third one is multiple sequence alignment. Multiple sequence alignment is nothing but when we have more than 2 sequence to be compared, the method is called as multiple sequence alignment. More than 2, 3, 4, 5, more than. Okay. And the sequence motif discovery in multiple sequence alignment. So from the multiple sequence alignment, when we are performing alignment among the amino acid sequence, so we can identify the motif, we can perform motif analysis. And the phylogenetic tree construction. So the phylogenetic tree construction is performed after the multiple sequence alignment. So the purpose of constructing phylogenetic tree is to identify the organism how much closely related to or distantly related to other organism. So that is the main purpose of performing phylogenetic tree construction. The phylogenetic tree construction, when we are trying to construct a phylogenetic tree, multiple sequence alignment is very, very important. Without multiple sequence alignment, we cannot perform phylogenetic tree construction. So, sequence analysis is very, very important for different purposes. Okay, we'll see about analysis method. What methods are used for analysis these sequence? Alignment. Alignment is nothing but to compare a sequence with the sequences that have already been reported or stored in database. So we can compare or we can align our own sequence, that is query sequence, with the database sequence in order to get information. It is simply, for example, uh, we are getting information from the unknown information. I mean, we are getting information for unknown thing from the known thing. That is the concept behind this. So the query sequence, when we are applying query sequence to the database has the tool for sequence analysis. The tool carries your query and it compare your query with all the database sequence, and whichever the sequence is closely related to your query sequence, it will show you as a result. So from the result we can get the closely related to our query sequence. Next, alignment can be global or local. When we are performing sequence alignment, it can be either manner, either in global manner or local manner. Global sequence alignment is nothing but alignment takes place for entire sequence is called as global sequence alignment. In the another case, local sequence alignment. Local sequence alignment is nothing but aligning the sequence in the specified area is called as local sequence alignment. For example, we are having two genomic sequence. We don't want to compare the entire genomic sequences. Instead, you are very much interested to align the particular portion of the genome or the gene present in the genome. So in such a case, we can perform local sequence alignment. The purpose of doing local sequence alignment, we can save the time, we can save the algorithm. Instead of comparing the entire sequence, we can compare the specified area. So when you are comparing the sequence at a specific area, the alignment is called as local sequence alignment. When you are performing for entire sequence, then it is called as global sequence alignment. So this is the difference between local sequence alignment and the global sequence alignment. These are the two different types of alignment methodology. Local sequence alignment is mostly performed to identify the gene and compare the gene present in the genome region or the database sequence. Global sequence alignment is performed when you are constructing a phylogenetic tree, or when you are comparing, or when you are performing the structure modeling structure, then it is performed the global sequence alignment. So in sequence alignment, there are two methods. One is local sequence alignment, another one is global sequence alignment. When the alignment process takes place for entire sequence, it is called as global sequence. When the alignment process takes place only particular region, is called as local sequence alignment. So this slide shows how local sequence alignment and global sequence alignment are performed. In the case of global sequence alignment, the alignment takes place for entire region from starting to end of the nucleotide. From the first nucleotide to last nucleotide, when we are performing alignment, then it is called as global alignment. Instead of first nucleotide to last nucleotide, we are concentrating a specific region of the sequence. Then it is called as local alignment. Local alignment is mostly performed for the gene identification or motif identification or the analysis of the codons in the genome or the analysis of ORF region in the gene or genome sequence. In such cases, we can perform local sequence alignment. Okay, so we have discussed about the sequence alignment, methods of sequence alignment, and the types of sequence alignment. Next to that, alignment tools. So for sequence alignment, we need tools. Without tools, we cannot perform any work in the bioinformatics. So whatever the work we need to do, we need specific kind of tools. That is very important. In the regarding that, BLAST is one of the most important alignment tool which is available in the NCBI. Already in the previous slide, we have discussed about the BLAST program. BLAST program is available in the NCBI and Washington University databases. So you can use NCBI BLAST or WU BLAST. The BLAST program was developed by Alfred, a great scientist. He developed a wonderful algorithm to search sequence in the database. In the NCBI BLAST, the BLAST programs are categorized into 4 different categories. One is nucleotide BLAST, second one is protein BLAST, third one is B BLAST, last one is T BLAST N. The nucleotide BLAST, the two major BLAST programs are nucleotide BLAST and protein BLAST. The nucleotide BLAST can be performed to compare the sequence or search the sequence with nucleotide to another nucleotide sequence. So this program, the nucleotide BLAST program, what it will do is, it will compare nucleotide sequence with another nucleotide sequence. So the comparison will be carried out between the nucleotide sequence. Whereas in the protein BLAST which carry a protein amino acid sequence as a query, the query sequence will be compared only in the another amino acid sequence. So that is called as protein BLAST. Last one is, sometimes we may have nucleotide sequence. If we have nucleotide sequence, we can perform nucleotide BLAST. Or sometimes we may have amino acid sequence. When we have amino acid sequence, we can perform protein BLAST. Sometimes we have nucleotide sequence, but we don't want to compare the nucleotide sequence with another nucleotide sequence. So instead, we are interested to compare a protein sequence. In such a case, the NCBI has another special kind of program called BLAST-X. BLAST-X receive nucleotide sequence as a query sequence, and it directly compares the nucleotide sequence with protein sequence. So the algorithm called as BLAST-X. And in another case, in the another situation, when we have amino acid sequence or protein sequence, we don't want to compare with another protein sequence. Instead, a translated nucleotide sequence we need to compare. In such situation, we can perform T BLAST N. Here T stands for translated, N stands for nucleotide. The translated sequence, that is protein sequence, will be compared with nucleotide sequence. So these are the 4 different BLAST programs available in the NCBI. In the previous video, we have discussed a lot about the applications of NCBI and the tools available in the NCBI. So BLAST tool is one of the important and frequently used tool for different kind of scientists. Those who are working in the genomic research or proteomics research, invariably all the research workers will be frequently using BLAST program. And algorithm. So already we discussed about the types of sequence alignment, local sequence alignment and global sequence alignment. The algorithm work behind the local sequence alignment and the global sequence alignment is, there are 2 algorithms. One is Smith-Waterman algorithm. Smith-Waterman algorithm performs local sequence alignment, and Needleman-Wunsch algorithm will perform global sequence alignment. For Needleman-Wunsch algorithm and Smith-Waterman algorithm, we can use pairwise sequence alignment. Or if you have two sequences to compare, you can perform Needleman-Wunsch algorithm or Smith-Waterman algorithm. Here, the Needleman-Wunsch algorithm works based on the genetic algorithm. So that is why all the databases are used with the Needleman-Wunsch algorithm, because it developed the Needleman-Wunsch algorithm developed based on the genetic algorithm. The error, the percentage of error will be very minimum for a genetic algorithm. So that is why the Needleman-Wunsch algorithm performed based on the genetic algorithm. Smith-Waterman algorithm is used to compare the sequence at specific region of the sequence, so local sequence alignment. So already we have discussed about the BLAST program and the tools are very important for sequence analysis, sequence comparison. BLAST program can be used in both way, both online and offline, and most of the scientists are using online tools only, because the NCBI is a famous nucleotides database. It contains BLAST program, so without any difficulty scientists are accessing BLAST program from the NCBI for the comparison of the other sequence or the retrieval of closely related sequence. So it is available in both online and offline. And another important search program is Conserved Domain Search Service, CD Search, Genome Workbench, Primer BLAST, Pro Spling and Spling. Pro Spling is for protein, Spling is used for the nucleotide search. Here in the NCBI database, we can see four different types of BLAST program. One is nucleotide BLAST, another one is protein BLAST, translated nucleotide BLAST. This is another special kind of BLAST program, that is primer BLAST. So primer is a short nucleotide sequence. So when we need to compare or search the similar primer, we can use BLAST program. Primer BLAST. Multiple sequence alignment. The tools for multiple sequence alignment is Embosswin, BioEdit, Megasoftware, and the ClustalW. So BioEdit is freely available standalone software. We don't need internet to work with it. And the ClustalW program is also available in both offline and online. And the specialty of ClustalW program is it is incorporated in the BioEdit program. Once you have BioEdit program inbuilt, you can use ClustalW. Mostly the ClustalW program is used for multiple sequence alignment. Once the multiple sequence alignment is performed, we can construct a phylogenetic tree. And most of the genomic scientists and the proteomic scientists are using a BioEdit program for sequence analysis, sequence comparison, and sequence editing. So all the work can be performed with the help of BioEdit. So that is why BioEdit is most frequently and famous program among the scientists, those who are working with the sequence analysis. Megasoftware, another sequence comparison, sequence alignment program, and Mega is specifically used for the construction of phylogenetic tree, because the Mega software the Mega software is inbuilt with so many genetic algorithms specifically for phylogenetic tree construction. Here also we can do sequence comparison, sequence alignment, and sequence editing. So this is the best example for how the two sequences are compared with each other. So this is a dot plot results. In the x-axis, one sequence is plotted, and the y-axis another sequence is plotted. Whatever the sequence present in the x-axis, when it is having the same sequence in the y-axis, there it is plotted a dot. So finally the dots are connected. Which kind of dot connects the diagonally straight line? That region has same alignment. So that is the significant result of dot plot result. So we have different type of alignment program, alignment method, offline tool and online tool. So the application is very important. Based on the application, we can select specific kind of tools, or specific kind of databases, or specific kind of format. So format is different, application format is different, tool is different, databases is different. So for our necessity, we can select database, we can select tool, and we can select the file format. So here multiple sequence alignment. The multiple sequence alignment is used to compare the structural analysis. Not only for structural analysis, it's also used for the functional analysis. The structural analysis and the functional analysis are two major important phenomena in proteomic research and also the genomic research. Those who are doing genomic research and the proteomic research, they will do comparative analysis and the functional analysis. So comparative analysis is to discover discover the defect and the effect of the sequence. Functional analysis to know the functions of unknown sequence with known sequence. Detection of relatedness. Already I have told you that the phylogenetic tree construction can be performed by multiple sequence alignment. And finally, the multiple sequence alignment can be used for motif identification. So these are the application of multiple sequence alignment. The multiple sequence alignment is often leads to the fundamental biological insight into sequence, structure, function relationship of both nucleotide and the protein sequence families. So that is why the multiple sequence alignment is considered as most important algorithm in bioinformatics. This is the example for phylogenetic tree. So this shows the sequence applied in the database. So for sequence alignment, so once the sequences are aligned, the sequence alignment is performed with a bioedit program, and the aligned sequence was stored in ALA format, and the ALA format is opened with Mega software. The Mega software is used to construct the phylogenetic tree. So here you are seeing the phylogenetic tree constructed based on the gene sequence with the help of Mega software. So here we have used two different software, a Bioedit and a Mega software. Bioedit software is used to align the sequences, and for the construction of phylogenetic tree, Mega software is used. So this is the best example for structural comparison, protein structure comparison. So already I have told you that the multiple sequence alignment is used for both functional analysis and the structural analysis. So it is the best example for structural analysis. So the protein structure can be compared either whether it is similar or dissimilar. If it is similar, we can go with some kind of work. If it is dissimilar, we can identify where the structural property is getting varied from one another. So with this, the application of multiple sequence alignment or the sequence comparison is over. So for this, I have used these references. Thank you, professors. Thank you all.